Greetings to all. Myself Gaurav Sharda, product manager from Abdos Life Sciences, and today we will be digging into a very fascinating and versatile technology, polymerase chain reaction or PCR. We'll be exploring how this technique has revolutionized the fields as diverse as medicines, forensics, and biotechnology. So, whether you are a scientist, a student, or just curious to know about the power of PCR, you are in here for a treat. This is for you. Today we have Miss Syeda Rushna, a brilliant and talented researcher from Jamia Hamdard. And to start the things off, we'll clarify first what PCR is all about. PCR is a molecular biology technique which is used to amplify specific DNA sequences. It is a cycle of cooling and heating to denature, anneal, and extend DNA strands that results into exponential amplification of the target DNA. Biotechnology as a field thrive on PCR techniques. It's the engine behind genetic engineering, allowing scientists to manipulate and modify DNA. This has far-reaching applications from producing insulin through genetically modified bacteria or engineered crop with desired traits. The possibilities are endless. So starting off, we'll first welcome Ms. Syeda Rushna. Welcome to the panel. So we'll start off with with our first question: that what is the role of PCR techniques in your research or your research studies?
Well, first of all, thank you so much for having me here today. It's a pleasure. Talking briefly about my research, it basically focused on exploring the host modulated action of microbacterial proteins.
Okay.
So, for that, we basically want to see and check how the protein, the microbacterial protein, it acts and modulates the host immune response for its survival. In a brief, we first basically select a protein with a broad review of literature and in silico analysis, and then we clone it, express it, and purify it. The first coming to the PCR, the first reaction, the first experiment that we perform in our research is cloning.
Okay.
So the amplification is the gene of interest, as we say, as the definition of PCR. So that is very important, and not just that. If we have any mutation production or knockouts or the knock-ins, so we use the same technique over there as well. So throughout the journey, it is a very simple yet very prominent technique that we use.
So what type of PCR technique you usually use in your experiments?
Uh,
Cloning or expressions?
so basically, as I'm in the initial years of my PhD, so we're performing cloning. So
Okay.
mostly we perform the conventional PCR, but in our lab we also perform the qPCR.
Okay.
So I think we have used both. We haven't used the reverse transcript PCR, but it is there. We are aware, but
Okay.
yeah.
So the reverse transcript is basically for the when you are working with
The,
virus.
where cDNA is
Yes.
proposed, mRNA wala.
Yes.
So that is it.
So what measures do you take to prevent any chances of contamination in your PCR experiments?
So, firstly, the very successful PCR product, the amplified product, is a backbone of any of the further experiments that you do. So, like, what are you going to do further if you do not have your amplified gene, if you do not have that? So, to have it to secure it, we have the most important thing that we do is to avoid the contaminants. The contaminants can be lurking around anywhere in your lab. That can be the previously amplified DNA that we were using, or just the cross contaminations if we are using different DNA at the same time. So that can be there. So there is a very golden rule to avoid the contaminations is to designate different areas for the PCR setup, for the pre-PCR and the post-PCR. PCR setup is basically when you're performing the reaction, when you're making the reaction. Pre-PCR is basically just using the PCR machine, and post-PCR is exposing the gel and all that stuff. So you have to designate different positions, areas for all of that, and do not ever mix or reuse or take the equipment or the consumables or the reagents that you're using in the post-PCR area to the pre-PCR area. That is the major cause where contaminations happen. Uh, for example, if you are performing the electrophoresis area should be definitely very separate. And the second thing is aliquoting the reagents. The reagents that we use they are like really important. Cross contaminations or contaminations can always happen over there. So you should aliquot from the main mix and have a small quantities of these very nicely, properly labeled. And then the most important thing is using the controls, the positive and the negative control. Negative control is where you do not use the template.
Okay.
And positive control is where you have the template and specific and all the things right. So we have this thing that in the positive control, we will get the result for sure. And in the negative control, we won't get the result because we are not using the template. So that would just rule out, the negative control will rule out if there is any contamination, and the positive control will rule out if there is any of the reagents are not working.
Okay.
So these are the things. And coming to the consumables,
Yeah.
always use filter tips.
Yeah.
So
to
avoid
prevent any chance of
yes,
cross contamination
or aerosols. Yes,
Exactly.
and the PP, the personal kit, that is the
equipments.
yes,
Yeah.
the gloves and all that
Yes.
stuff. That should be, the area should be sterilized. So that's all the used
Okay. Thank
steps.
you. So what things do you consider into account while choosing the right PCR product?
So there are basically different parameters that we take into consideration while choosing our components. The first and the foremost and a very important one are the reaction components. They play a very crucial role in PCR. Like, for example, we have major PCR components such as the DNA polymerases, our enzyme, and we have the DNTPs, the cofactors, the NFW, and the template of the DNA. And in case if you are performing the qPCR, we have the probes or the dyes. So it basically, if there is any contamination or any degradation in the components, so we won't be getting our desired product. So that is very important, firstly. Second thing that is that we use are the consumables.
Okay.
So now it goes two different ways. If you are doing the conventional PCR or if you are doing the quantitative PCR. For the conventional PCR, we have the tubes. So the PCR, if the conventional PCR is done in the PCR tubes or it is done in the stripes, so we have to check, we have to literally maintain and see and ensure that there is a thermal stability, like the plastic that is used is thermal stable, so as to ensure that the inside the temperature is maintained throughout. As you have already mentioned, it involves the denaturation, the annealing, and the extension. So we need different temperature for all of these processes. The temperatures varies about from 95 degrees Celsius to 50s.
Yes.
So it should be thermal stable. So secondly, if we are performing the qPCR, so it is a little bit complex and different from the conventional PCR as it involves the fluorescence labeling to quantify the amplified DNA at different period of time.
So do you think there is a need of innovation in the PCR product lines that can improve the efficiency of the PCR experiments?
Yes, definitely. There is always scope for improvement, and innovations are always welcome. That is make it us easy to perform the experiments. So as I have been already talking about the thermal stability, so the first and the foremost is that we want the products that we are using are thermal stable. The second thing that I had been mentioning was the cap. We want to ensure that there is no contamination, cross-contamination while the samples are in the cycler, or there is no evaporation because that will cause the loss of our sample and we do not want that. And so the proper sealing should be there. And the third is to ensure that if we can have a system where we can have all the products and all the steps combined in one single machine. So integrated system can be used if it is possible. So yeah, I think these three are the major innovations that can happen. Because in our lab, we have already experienced one episode where the tubes lost their shape when they came out of the cycle, and that was very disheartening because we lost our components and we lost our results. So that the two major ones that I have been using and the integrated system. So yeah, that's what I would say.
All right.
Thank you.
Yeah, so, well, thanks so much, Ms. Syeda. It was lovely to have you on the panel.
Thank you so much for having me.
Yeah. And well, that wraps our exploration of PCR applications. I hope our viewers find it enlightening as I did. By addressing these questions, I hope the viewers can understand the use of PCR applications and how they can use the PCR consumables effectively in their experiments. Thank you.